Hey everybody, in this video I am going to go over the mechanism of class switch recombination, and this is the mechanism that can take a B cell that makes, say, the IgM and the IgD isotopes of antibody and convert it to a B cell that makes, say, IgA antibody or an IgG or IgE. And as we'll see in this video, there are specific subclasses of these. So we might, instead of getting IgA, we might see IgA1 or IgG3. So I'm going to cover that in this video. So I'm going to start with a diagram that's very similar to diagrams I've used in previous videos. And this is a diagram of an IgH locus that has already undergone VDJ recombination. So we have a V segment joined to a D segment joined to a J segment. And in previous videos, we saw how this rearranged IgH locus could be transcribed into a messenger RNA that allows for production of the IgM or Ig delta antibody, depending on how this piece right here was spliced out. And if you want to review that, I would check out the previous video to this one. So now looking at this locus, I'm adding some more details here. I added some more constant regions down here. So all these blue squares here, these represent different constant gene segments. Now I also added some more detail that I didn't include in previous diagrams of the IgH locus. And that would be these blue circles right here. And these blue circles are going to be found upstream of every constant gene segment except for the delta segment, except for this one. And what these are called switch regions. So the switch regions are about 1 to 10 kb long, and they have lots of sequences that I should say lots of intervals that have lots of this sequence. Let's see, A or T, this would be the 5' end, A or T, followed by a G, followed by a C, and then followed by another A or T. Ooh, did I say A or T? I meant A or G. A or G, followed by G, followed by C, followed by an A or T. So lots of these sequences are found throughout these 1 to 10 kb switch regions. And these are important for class switch recombination. So how does this mechanism work? So let's say the cell, for some reason, receives some signals and it's being induced to switch from the IgM, IgD isotypes, right here, a cell that produces IgM and IgD isotypes, to one that produces, say, Ig alpha 1. So what will happen is these switch regions here will be broken. So double strand breaks will be placed in these two switch regions. So it's going to look something like this. So after the double strand breaks are placed in those switch regions, this whole piece right here, this whole interval is going to be lost. And then this, so the 5' end or the upstream side of this switch region here is going to be joined to the downstream portion of this switch region right here. So we're going to get something that looks like this. Now, so this part right here is the same down here Now remember, this is now the coding region for the variable portions, right, of the heavy chain. And this segment right here has been removed, and I moved this alpha 1 segment up here. Alpha 1's up here, and I put some other gene segments in here, so they would be off the page up here, and so I just move them up here so we can see some of the others. So we have gamma 2, gamma 4, epsilon, and then there are even some more down here that I'm not diagramming. So these are going to be joined, and essentially they're going to be joined by non-homologous end-joining proteins. And remember, this is a highly mutagenic process, but it doesn't matter. It doesn't matter because when these are linked together, it's not coding for anything here. This is part of an intron that's going to be removed when this area is transcribed, and that transcript is translated into the heavy chain for the IgA1 isotype. So let me show that down here. As we've covered over and over again in the last previous videos, each of these segments, variable segments, has its own promoter, but the one closest to the D and J segments is the only one that's active. When it is used to transcribe an RNA, so that RNA polymerase is going to transcribe everything down here, and it's going to stop at the end after this gene segment right here. So assume that each of these gene segments has the termination signals that cause RNA polymerase to fall off the template. So that pre-messenger RNA is going to look something like this. You know, I put the poly A tail in, the 5' cap, but I wanted to show you is that the 5' splice site for the primary intron is going to be here right after the J segment. The 3' splice site is going to be here right before the alpha 1 constant gene segment. And this whole thing, we're going to call this the alpha 1 intron, and that's going to be removed by the spliceosome, giving us this messenger RNA that could be translated into the heavy chain protein, 2 of which are included in each antibody. And because the constant gene segment is the alpha 1 type, this is an IgA1 isotope for this antibody. Now note that the variable regions were unchanged by this class switching process. So IgM and IgD, which were the antibodies produced by this cell before it underwent class switch recombination, they recognize the same antigen as this IgA1 antibody, which is now formed by the cell after class switch recombination. Is there anything else I wanted to address? So how do the double strand breaks occur? Right? How do they occur? So let's take a closer look at the switch regions. So they can be from 1 to 10 kb, so a wide range of sizes. But like I said before, they have these 4 base motifs like that occur frequently throughout the switch regions. And so A or T followed by a G followed by a C followed by, A or G followed by a G followed by a C followed by a A or T. And I've, came up with just an imaginary switch region, and I'm sure there's a part of a switch region in humans or mouse that looks something like this. They're pretty big and they have lots of these motifs. So I've shown a small region with just 5 of those motifs. So here they're in the 5' to 3' orientation. That goes from left to right, and here they're going from right to left. So here's our AGCA, AGCA. So they're inverted relative to the motifs in the top strand. So how do the double strand breaks occur in the switch regions? So when class switching is needed, an enzyme called activation-induced deaminase is expressed, and this is the same enzyme that is thought to participate in somatic hypermutation. So we're using, this is the second time this enzyme has been involved in sort of antibody functions, or 2 different roles for aid in forming antibodies. So, and what AID does, remember, it has a preference for these sites right here. It prefers these A, G, C, well, A or G, followed by a G, followed by a C, followed by A or T. It prefers these motifs, and these motifs are found throughout the switch regions. And what it does is it changes the C's to U's, as we saw in the previous video. So please check out the previous video so you can see some diagrams of cytosine and how it changes the cytosines to uracil by deaminating the cytosines. And okay, so that leaves us with a lot of UG mismatches in the switch region where the double strand break is going to be placed. Okay, so how does this lead to a double strand break? Well, the most popular model is that base excision repair, which we've already learned about. What it does is it goes through and it cuts out all the U's using the DNA glycosylase, and it puts nicks in the DNA backbone using the AP endonuclease, and it gives us lots of nicks in the switch region. And why are there lots? Well, there are lots because there are lots of mismatches in this region. And the idea is that because these nicks are so close to each other, they're essentially they're essentially causing double strand breaks, right? So the nicks aren't directly across from one another, but they're so close that what we're getting is a staggered double strand break. And there's so many of these nicks on both strands that essentially we're getting a double strand break in the switch regions. And okay, so that allows double strand breaks to occur in each switch region. And non-homologous end joining can then repair it in such a way to put that alpha 1 gene segment directly downstream of the joining segments to allow the cell to make the Ig alpha 1 isotype. Now it could also do, if the cell needed to make epsilon, well, it would put the break here. If it needed the IgE isotype, if it needed the IgG2 isotype, it would put the break here. So there are some things we're not covering. I don't actually know the answers to the questions I'm about to go over with you, but I don't even know if researchers have figured this out yet. So one question, how does the cell restrict AID to the switch regions, right? The cell probably doesn't want mutations throughout the genome. So how does it restrict the enzyme to placing mutations within the switch regions? Also, how does the cell control the switch from the IgM, IgD isotope producing cell to a specific isotype? Like, let's say that the cell receives a signal to switch to IgE. How does it control which switch regions are broken? So which switch region, so it's always going to need to break this one, right? The one upstream of the mu gene segment. But how does it know to, direct AID towards this switch region and not any of the others? So it's possible that maybe the switch regions are masked, maybe they're in a condensed chromatin and they're only accessible when cells receive certain signals. Say a cell receives a signal to switch to isotype IgE. Well, maybe the switch region near IgE becomes opened up for AID while the others remain in condensed chromatin. So what are the factors that would be involved in opening that specific switch region? I don't really know. I don't know if that's how it's done, but it might be possible. So we're not going to worry too much about that, but there's lots of interesting stuff that we could go on forever, probably study molecular biology aspects with respect to the immune system and antibody production. Plus, we never even looked at how the T cell receptors are formed, and that involves also VDJ recombination. I'm assuming the processes are similar. So this is going to end our segment on molecular biology in the immune system. We will be looking at gene therapy and things like that in the future, but I think we're done with antibodies for now. Okay, so if you have any questions, let me know. Send me an email, or I'll see you